Saurav, can you hear me?
Yes, ma'am.
Okay. Are we recording this meeting?
Yes, ma'am. Yes, ma'am.
Brilliant. So what I'll do is I'll open both of my machines start so I can see the chat box also, and I can also see the main screen where the slides are. Good afternoon, everybody. Welcome to the intro to Unix, or the Unix 101 for the high performance computing that we have at NIPGR. I see that we have representation from almost all the labs at NIPGR, and we also have some people from outside of NIPGR. What I would like you to do, if possible, please can you put your name and your current role, if you're a PhD student or a postdoc or a PI or a staff member. If you could just put it in the chat box, then I will know and where you're from. I mean, we know the NIPGR people already, but if you're not from NIPGR, you can put the name of your institute as well. So while you do that, I would like to welcome you once again to the introduction to the Unix command line interface uh, interface course that we are running as the first absolute basic course here at NIPGR. The idea was originally to give you a course on the phytocluster, on the high performance computing itself, but we realized that a lot of biologists, including you and many of my own students, are still, primarily biologists. And so programming is something that is not happening on a daily basis in the lab, and you've not even learned it. So I don't intend to teach you programming essentially, but some very basic ways of interacting with the computer, especially when and if and now that most of us are generating a lot of data that needs to be assessed in different ways by using softwares and programs. So hopefully this course will apply to you regardless of which lab you are in and what you are doing. Okay, so the first thing we do is to welcome you to the phytocluster facility that we have now made online and active at NIPGR within our phytocluster. It is being handled jointly by myself. I'm Gitanjali Yadav, for those of you who don't know me yet, and my colleague Shailesh Kumar. Both of us have been at NIPGR and are handling the computational and bioinformatics facility here. So within the phytocluster, we have several different components. Today is just a day to tell you how to, today is a simple course to begin on Unix, on how you will access all of these different components. But eventually, when you learn Unix and when you start to become a little bit more comfortable with it, we will ask you questions like which one of the components do you want to access? And then we will give you access to those for doing your compute work. Right now, today, you do not need access to any of our compute or archival storage or cloud components, which you can see on the screen over here. Today, you are all within the PhytoLearn Abhyas idea. The PhytoLearn Abhyas itself is a server, and we have a portal through which some of you, those who are within the NIPGR LAN, we will ask you to log in to us with that. But that is also not critical because this is a very, very, very basic course and it is only about simple Unix commands. So you can even just run it on your own laptop, device, desktop, whatever you have. But eventually, for the intermediate courses, as well as for the high performance computing courses, you will be required to use the Abhyas training portal. And that is done through remote login. And so remote login is something that we will teach you today. So unless you have learned remote login, obviously you cannot do remote login, right? Which we will do through SSH eventually. So before we begin the whole idea today, I would like you to just ensure that on your computers or your laptops or your devices, you currently have, a Unix terminal open. Now, I had written an email to you that some of you may not have a Unix laptop or access to a Unix desktop. In case you have a Windows machine, the Windows machine also has an app called the Putty app, right? And so you can use the Putty app, but there are other things you can do on a Windows. You can download some apps called the MobaXterm app, which I had sent the link to in my email in the morning today. You don't have to rush into doing it right now, but that is something that will easily allow you to access the phytocluster, especially from within NIPGR. You don't need to install anything. You just need to have a terminal Through the terminal, we will give you access to the entire phytocluster, the storage, which we call Beej, the multi-compute node, high-performance computing unit, which we call Vriksha, the deep learning modules called Mool, the GPU and very high compute intensive, graphics cards, which we are calling Agni, and of course the on cloud server that we have for synchronizing your lab data with the hostel data or the home data or the phone with the desktop and so on. Smaller sized files get synchronized on the Vyom cloud. The larger file synchronization and backup happens on the archival storage, right? So if you have a Unix terminal on your laptop right now or the desktop through which you are connected to me, please put a yes in the chat box, and then I will know how many of us are. I'm going to be looking at the chat box to make sure that you do have it. So very nice. Very nice. Welcome everyone. I see Janvi, Radhika, Mustafa, Rinchula, Rajarshi. Brishti is joining us from NII. Welcome, Brishti. There's, of course, Zainab, Ashish, Shankadweep, students who I know. Very good. Is there anybody who does not have? Okay, I asked the wrong question, and in future I will make sure that I ask the right question. So when I ask you to type a no, that allows me to see who needs help. So as of now, I see everybody does have the Unix terminal. So if you have the Unix terminal, can you simply type some kind of a command on it, such as ls? Let us say ls is like a listing command. And you can see on the screen also right now, I have typed ls right here. So do you get any output when you type ls on your terminal? Does it give anything or does it not give anything? Let's see in the chat box what you guys have. And I especially want to see a message from anyone who does not have the terminal, because that is an important thing. And one of us will have to come and help you in case you do not have the terminal. So let's do that. And of course, in case you do not have the terminal, do not worry. We are going to help you with that. So that is another important thing to set aside now that we are looking at the data. I'm looking at I am going to go slowly. I have a habit of going too fast, but I'm going to go slowly today so that the slowest person in the room, who probably does not understand everything, but is unable to or unsure about telling me so, please feel free to type in the chat box in case you do not have a terminal access. Also, tell me what is the output of the ls command. You can write a few lines or just a few words. What do you get when you type the ls command on your terminal? Is Shashi here also, Saurav?
Yes, ma'am.
तो शशि से एक बार पूछना, is it possible for you to message me or text me?
Hello, ma'am. I am here only. I will reply to
Great, great. Shashi, can you text me a mechanism to online access the Learn Abhyas? Because then I can type these commands saath ke saath and people can see me rather than just showing the students the screen or the PowerPoint slides. I would like to open
Yeah.
the.
So just I am typing the URL. So if the
नहीं,
anybody
सिर्फ
has
मेरे लिए बिकॉज बाकी स्टूडेंट्स को हमने सबको एक्सेस दे रखा है, तो आपने सिर्फ मेरा वाला बताना है अभी। ये मैंने क्या क्या इसमें और ओपन कर रखा सब। तो आप मुझे बताओ, आई थिंक इट विल बी training.nipgr.
yeah training.npj that is okay, but that you need to change your DNS
I think
settings.
that's there. I think that is all changed. Sort of did it. So it's been done
Okay.
for me.
Training 1.
No, let's start the last one. Let's take the last one.
Okay.
Shuru wale, I think, are with the other students today. We've given
Just put
notice.
HTTPS in above that first before training. You just type HTTPS.
Ah, yes, of course, of course. And this is what others will also do, those who have their own training logins. Hai na? So after HPC, should I type something after HPC also?
No, nothing.
That's it. Should work.
It's not working. Just a minute.
Mhm.
Last IP address of that.
हाँ बता दो. Yes.
64. 172.16.
Yeah.
21.64.
Is that correct?
Yeah.
Okay, let's see.
Sorry, just you. So go to previous one. I forget to tell you the port number.
Yeah, okay, okay, perfect. All right. Training 14
Dot
dot
NAPG address.
NRPGR dot HPC
Port is colon 9090.
9090.
That's all.
We try an HTTPS and let's see if this works. Okay, it's not secure, but I think we should be able to still go to it because it's our own. Let's see. All right.
Here you
So,
can log in with ST1, ST2, ST3.
ठीक है, I'll just try the last one, I think.
In below your left hand side, there is a terminal.
So this is what everybody has to do. Those of you who have logged can do exactly what I'm doing with your user ID and your password and pressed terminal. And so I'm on the terminal right now. Okay. So this goes with my screen here that I'm presenting also. I think I'll just make a little bit bigger presentation. Is there a better way of presenting this? Chalo, theek hai, aise hi rakhte hain. So I'm going to type out ls now. So here I am on the command line and I'm typing ls, right? Let's see what I have. I just have R here. That is my output. Did anybody tell me what you guys got when you did the same thing today? I mean, when you accessed your own terminal and typed ls? Okay, Sankhadweep says that they cannot type into the portal through SSH. It is showing something is not working. So can you try, Sankhadweep, exactly what I have done? That means you use the SSH portal link on your browser, whether you are using Chrome or Safari or any browser. Just go onto the browser, but you have to be within NIPGR to do this. And Rajarshi has got an output, and Janvi has also got a whole list of files. This is all brilliant. Very good. What about the students outside NII? Are things okay with you guys? Brishti? Okay. No, you don't. Mustafa says the SSH should work. Yes, it should work like that, Mustafa, if that has been given to you. But in case it is not working, one reason could be that you did not fix your DNS, primary DNS. Did you change your primary DNS on your computer, Mustafa? Or did you meet Renu earlier before this meeting so that you could check that the login is working? You can type your answer or you can speak whatever you feel comfortable. Okay. So Mustafa,
audible, ma'am?
yeah.
Okay. Okay, I'm trying to change.
So Mustafa, you have to change your primary DNS. Only then will you be able to log in or perform a remote connection to this. But do you have any simple Unix terminal on your machine already? Because there also you could just type in an
Actually,
ls.
ma'am,
Huh?
actually, I have Linux system already.
Oh, very
So I
good. So you
just,
don't need brilliant. So just open your terminal and type in ls, and that means ls should be working for you, right?
yes, ma'am, but I'm unable to log in from this
So
SSH,
yes.
this
So
training12.nipgr.hpc.
yes, for that to work, you have to meet us sometime and get your primary DNS sorted. Does that help? So right now you
Okay.
cannot do it because we are already in the meeting. It had to be done before the meeting. So when I wrote an email in the morning, the email was mainly to tell
हाँ,
you कि meeting से पहले ये सब sorting out कर लो. पर कोई बात नहीं, अभी आपके पास Unix है, so you just use your Unix and let's go ahead. Right? So Shashi has put a message in the chat box that you have to change your primary DNS settings. If anyone knows how to put the DNS settings, only then it will resolve the host name for you. The host name resolution, resolving of the host name like training.hpc or anything else basically happens only when you when you use the DNS, which is the domain name server. So if you've not enabled the DNS, then you will not be able to resolve the host name. So don't worry about it. This had to be done before the training course, but now we are already starting the training course. You don't have to do it right away. In case some of you feel compelled that we do not have any Unix at all with us, and so we do need it. In that case, you can just change your DNS settings and get into this. Okay. Is there anybody who does not have access to the terminal still? Because I would like to see who it is who does not have access to the terminal. So Rakesh has an interesting question. Rakesh says, does the ls command tell you where you are? No, Rakesh, it does not tell you where you are. The ls command is a listing command. It just lists wherever you are in the directory. It gives you a list of the files in those directory, exactly as Baljinder has put in the chat box. But that is a very good question. You should generally think about what are these commands doing? And the Unix commands are actually very simple. You can generally just get an idea about what that command is doing. Okay, so let's again go back to the fundamentals and come back to the main screen here. While you are typing out or letting me know any other issues that you have with your Unix setup, this is just for setup. Okay, so in the first basic setup stage, I'm expecting that all of you will have a terminal. You could have a terminal on your own computer. You could have a terminal through the PhytoLearn Abhyas portal, and you could also have a terminal through Putty. Any of these terminals is completely okay. Some of you who cannot resolve the host name are either outside of NIPGR LAN. You have to be within the NIPGR LAN to access to resolve the host name. And if you are within the NIPGR LAN and you are still on campus not able to resolve the host name, it means that you have not changed your DNS settings. You have to get the domain name server open. Once your domain name server settings are fixed, then all the domain names are going to be recognized by your device. For example, यहाँ screen पे आपको दिख रहा है, there is a domain name called Beej. So it will be the storage of NIPGR will be accessed by beej.nipgr.hpc. Similarly, the GPU can be accessed by typing agni.nipgr.hpc. Similarly, the in-house cloud that we have at NIPGR is accessed by vyom.nipgr.hpc. But for you to resolve these host names, for your computer to resolve these host names as a real computer in the server room downstairs, you have to activate the domain name server settings. So you have to change your domain name server DNS settings on your computer. This can be done either yourself if you know how to do it. If you don't know how to do it, please come to us, meet us in the office in the CBBF. Our staff members here, Subhashish and Saurav, are there in the server room all the time, and you just bring your device downstairs, they will change your primary DNS for you. और जैसे ही आपका primary DNS activate हो जाएगा, you can absolutely start to resolve the host names. Okay, is that clear to everybody? Can I have a little yes in the text boxes to understand that this is clear to everybody? If it is not clear, please come and meet us in the CBBF after the meeting. Thank you, Asim, Avni, Ruby. That's brilliant. So moving on about, what is the Unix environment essentially? The Unix environment is a core programming environment for HPC. It is not a language, it's an environment in which you work with different languages. So high performance computing systems are built majority on Linux or Unix only. And therefore, it is very important for you all, in case you want access to the high performance computing environment that we have at NIPGR, or even in your own institute. NII has a lot of high performance computing servers. So in case you want to use any of such high performance computing system, you need to have a basic working knowledge of Unix. And this is essential for I'm not saying you have to know a programming language. A working knowledge of Unix allows you to access these HPC systems. And it is not enough to access. You have to be able to navigate also कि मैं कहाँ पे हूँ, कहाँ जाना है. For example, just now a very good question was asked by somebody that does the ls command tell me where I am? So the ls command will not tell you where you are, but the pwd command, which I will show you by and by, the present working directory will tell you where you are. Basically, it is your present working directory, right? So a working knowledge of Unix is therefore essential for accessing and for navigating the high performance computing systems, as well as for running jobs. Now, when we are giving you access to such a huge unified environment, it is very important that you know how to run these jobs, right? And whether you run the jobs nicely, or whether it takes you time to run the jobs, or whether you have to learn programming to run those jobs, is a separate issue. But just now, even to basically access, you need to be able to have an essential working knowledge of Unix. And therefore, this course that we are doing today is for you to have this essential basic working knowledge, okay? And the example that I have used over here is that of a scholar. Now think of a scholar. I have one of the students in my lab, Mansi, and imagine Mansi as somebody who is, kahan gaya Mansi ka example? So you could be, anybody here. Let's say Preeti is here, Kamakshi is here. So you're a scholar, you're collecting a lot of DNA samples, and you're working with proteins or mass spec data, and you're collecting hundreds and hundreds of samples ka data sets. Or if you're doing genomics, then you're collecting not just hundreds, but thousands of genomics, transcriptomics, or other regulomics data files. And to work with these files, it is not simple. You cannot manually be looking at any of these files. You have to be able to navigate these files, look at them together, sometimes combine them, sometimes filter them, sometimes try to know what I need to do with them, and so on. So wrap your head around them. And therefore, it is very important for research scholars, particularly in biology, to understand or to have a working knowledge of the Unix system. Usually all of you are already working on computers, all of you are already using a lot of chat GPT. So I'm sure that you have very good, comfortable working essential knowledge of the computers, right? And generally what happens when you're working with a computer, you're essentially interacting with it in various ways. All of you are very, very familiar with the graphical user interface, or the GUI. Do you see on the right side? That is the way you're probably on your machine. When you open a folder, it is visible like that. There are some directories, some folders, there are some files, and all of you are very familiar with this right side, the right hand side wala picture. The same exact information. What is the information on the right side? Does everybody recognize the right side picture? It's a simple GUI interface of a folder. So we are looking at the data shell folder on the right side right now. Can anybody tell me how many directories are there inside the data shell folder? Just by looking at the right side image, you can tell me how many directories are there, right? And if I ask you what are the names of, very good, Baljinder, there are 3 subdirectories. Radhika is absolutely right. And how many files are there? Files meaning not directories. So directories contain files, and files are individual. Very good, Avni. There are just 2 files. So you know there are 5 things sitting there. 3 are folders and 2 are files. I also see Sankadweep has written a message that he changed the DNS and he's in. Very good, Sankadweep. So you have resolved the host name. Brilliant. So this is exactly, it's a matter of a second or a couple of maybe less than a minute that you can change your primary DNS settings, and you can access the Phytolearn Abhyas. And in the same way, you can access the GPU, the CPUs. You just have to tell us or fill a small form in which you say, okay, now I have taken the training, and now I would like to access the compute of the Phyto cluster, and I would like to use this, or that. And we will provide access exactly through the same DNS system. Okay. So very good. Mohit is absolutely right. There are 2 files and 3 folders. Now, the same information can also be accessed on a text-based interface mechanism. And the text-based interface mechanism is the one that you're seeing on the left side of this slide. And on the left side, ka matlab, where you have an example of the same exact folder, which is the data shell folder, and it contains 3 folders and 2 files. Can anybody tell me, see on the right side wala image on the GUI, you easily jumped and you were able to find ki kitne folders hain or kitne files hain. On the left side, it is just text. Can you still tell by looking at the text that this one, there are 3 directories and 2 files? नाम तो सब पहचान गए, सब same है, coronavirus, molecules and sequencing. But do you also see that there is a way to identify who is a directory and who is a file? And if you do, if you can guess what it is, type it in the chat box. Otherwise, I will tell you the answer. Brilliant. So Rajarshi has a very interesting answer. Rajarshi says that all the files have a TXT extension. But Rajarshi, these are just text files as a random event. Files could be so many others. There could be Word files, there could be PowerPoint files, there could be, Excel files also. Your data can be in any format. So it is not necessary that if it is a file, then it should have a TXT. There is another trick here. There is something else here which lets you in one go look at whether or decide whether it is a directory or whether it is a file. Now, Asim has given one answer, and Asim says that if the if the output of your command ls minus l listing command starts with a D, then it means a directory. And if you see, and Baljinder has something like file size also, if it is 4096, then it is a directory. Okay. So Rajarshi has a second point that the color codes are interesting. Yes, the color codes are often very clear. The first thing that we can do is the color code, but not all of your Unix layouts, all of your terminal layouts may not show the color. See, my terminal shows me these colors. So if I have a directory, it is always showing up in blue color. If I have files, they show up in green color. But maybe, just maybe, your terminal, when you are accessing, or the phytocluster terminal, which we will together access, may not be showing this kind of a directory structure to us. In which case, the right way of identifying who is a directory and who is not a directory is usually the first letter of the listing. Here you see the listing, and does this whole line, every row is basically the name of a document, whether a folder or a file, just like in the GUI interface on the right side also, isn't it? So if it starts with a D, then it's a directory. If it starts with a dash, then it is not a directory and it is a file. Okay, the main thing right now which I want you to note is that you can get a view of the exact same data that is in your usual GUI interface, also through the text-based interface that you have on Linux or in Unix. Okay, that is the important bit I want you to know. So Unix is basically a family of operating systems such as Linux, such as the Mac OS. I'm talking to all of you today from a Mac OS system. And the Unix operating systems generally have a program or an app or a mechanism that allows you to interact with the computer using a text-based command line mechanism. So that program or that interpreter or that command line interface is basically your terminal. And that is the terminal you are seeing on the left side right now, the black colored background box on the left. And this command line interface or this terminal is basically talking to you through a command line interpreter. Okay. And this command line interpreter is what we call a shell. And that allows you to interact with Unix-like operating systems. And one of the most famous kinds of shells is the bash. And the bash is the one that we will also be using over here. Here is Mansi's example. So Mansi is a forest ecologist, and she has just returned from a survey of the Corbett Tiger Reserve, where she had been sampling the understory of the forest, particularly soils, because she wanted to understand what kind of microbial and fungal life forms are there. So Mansi brought back 1,000 samples, let us say, over the last couple of years, and she has run all of these through assay machines or genomics machines or relative abundance machines, and she has probably identified 300 proteins that she wants from these samples, from each of the samples. And now what she has is 1,000 files. So imagine all of these 1,000 files Mansi has to put through a program. Let's name the program stats.sh. Okay, it's a shell script program. How is Mansi going to run the 1,000 files on the program? She's going to take each file and run it on the program individually. Is that a good idea? So that is a huge task on its own. But that is not all. Mansi has to take these 1,000 files and then she has to run the program on the 1,000 files. And then her supervisor, somebody like me, says you have to write the results by the end of the month, madam. And you have to have your paper. And the paper has to appear in a special issue of, Corbett letters or something. And you have to get this done immediately. So if Mansi chooses to run the program by hand using a GUI, remember the other side, that easy right side picture where you have a GUI, Mansi will have to select and open 1,000 files one by one, or har ek pe she will have to run the program. And if the program takes, let us say, roughly 30 seconds, usually some programs take longer, but if this program, let us say, takes 30 seconds to run on one file, then do you know how long Mansi will take to run all the 1,000 files? Assuming she's not eating, having any chai, talking to any friends, just sitting on the computer and clicking the GUI, click after click after click every 30 seconds. So this is going to be a very difficult task for any researcher like Mansi or yourself. But if you have the shell, if you have access to Unix, and if you have a very basic working command line structure information or essential knowledge, you don't need to write the code. The code is already there for you. It is called the stats.sh program. All you have to do is you will assign the task of doing this mundane job of ek ke baad, ek ke baad, ek ke baad, ek to your computer, so that Mansi has more time to focus on the attention of writing the paper that she has to write. Okay. And this is what each one of you also needs to do. Does that make sense? I don't see any questions in the chat box, so I'm going to assume that this is okay for you guys. So for Mansi to achieve the task, Mansi, what she needs to know is, number one, how to navigate to a file or to a directory in her machine. Also, how to create a new file or to create a new directory. And then to check the length of the file. That also Mansi needs to know. Are some files very, very long? Are others shorter? How many files are there, and so on. And she has to know how to chain a couple of commands together, right? So once she has chained those commands together, she has made something that we call a pipeline. And the moment the pipeline is ready, she has to retrieve one by one each one of her 1,000 files and push it to this pipeline. That means she has to iterate many times or 1,000 times over the pipeline. And then she will run a shell script which will contain her pipeline. So this is the task that she has to do. And this can easily be done on a Unix terminal. And that is what you need to learn also with your little tasks, with the tasks that are repetitive and you do them again and again because you have to click, click, click on the GUI interface. You can save yourself all the clicking effort by knowing very basic essential working skills of the Unix command line interface. Okay. So Mansi's example is just given to you to basically think about yourself. What kind of data do you have? Can I have an answer from you guys? Do any of you have multiple files that you're working with? You may have text files, you may have Excel files, you may even have, I don't know what kind of data. Do you have FASTA files that you're working with? Or are you having mass spec files? What kind of data are you looking at in your lab, in your work? And this will give me some idea about the kind of stuff students are doing. I expect, because we are in the National Institute of Plant Genome Research, a majority of you all will have something like, genomic sequences and genomic data or transcriptomic data or any other kinds of omics data. But I'm very keen to know if any of you have other kinds of data also, and how do your FAST files look? So Ruby has FASTQ files, Avni has whole genome sequencing data. Again, these are genomic data set, very good. What do the students at NII, what kind of data have you ever worked with? Even if you are like very new students, fresh PhD students, did you look at any kind of data during your master's thesis? Were you just putting all of your data in an Excel file? Even that you can put, and you can say all of my data is in Excel sheets. That is good enough for me. At least I'll get to know what kind of data, what kind of formats you guys are looking at. So, of course, Ashish is looking at mass spec files, and he uses a lot of FASTA files for the protein sequences that are coming out of the mass spec. Very good, Ashish. And do you use GUI, Ashish, to look at your files, or do you use command line Unix terminal to look at the FASTA files? You can type your answer in the chat box. But yeah, so GUI. So I'm hoping, Ashish, that you and some students, so you are the example instead of Mansi's example. I think I should give your example, that I'm hoping after this course, you will at least feel a little bit interested in looking at the text side also. PDB, Rishabh, very good. You have CSV files and PDB files, brilliant. That is a lot of data comes like that. Absolutely correct. So moving on, I just told you about, the Unix shell. So here are 5 reasons that allow you, that ensure that you fall in love with the command line. So what is the command line interface, and why would you as a researcher, why should you as a researcher use it? First of all, the shell is basically an interpreter, okay? And you can just, it allows you as a terminal. If you open the terminal, you can read, you can type, and you can run a lot of programs doing things together. And in this course, we are using the default shell that comes in most Unix environments, implementations. But from the time that I started doing Unix 30 years ago, I was mostly using the C shell or the TC shell. The bash is something I started using in the last decade only, but the bash has become the most common command line interpreter now. Programs of any kind that you may download or be shared, be given to you by your supervisor or your friends or your batchmates or your lab mates, or those that you get from collaborators, can be run in bash by simply entering the command at the command line prompt. Just like the way you typed in the ls command right now. Don't worry about it. We will come back to it later. Right now, I'm only telling you what the terminal does, right? And the main advantage of the shell, therefore, is its very high action to keystroke ratio. Like in the GUI, the action to keystroke ratio is crazy. एक keystroke पे एक ही काम हो पाएगा. But the action to keystroke ratio completely changes for the shell or for Unix, because एक ही command पे, एक ही keystroke पे, you can run hundreds of actions, okay? And that is the main thing why you should use the command shell. And another main reason why you should use the command shell is that it is supporting automating of the repetitive tasks, as well as finally its capacity to access network machines. So NIPGR Phyto cluster, even the NII high performance computing cluster, all of these machines are machines on the network, and they are networked machines. So you can't just go down to the server room and think, I will switch on the button on off karke and I will access it. So you have to access these network machines, and it is not possible to access these machines on the GUI. There is no way you can click the button and say, I want to access this. The only way you to access a networked machine, such as a high performance computing system, is to do through Unix. And that is why you need to know how to use the Unix shell. A significant challenge that a lot of students, and I'm expecting you will also have, about using, or first time users who come to the shell, is about knowing what command should I run and how should I run those commands. Most students, 95% of you, if you have not taken this course, or if you just, if you are just pushed into running using the Phyto cluster or the high performance computing system, you will learn it eventually, but much later, and you will make so many mistakes before that, and you will also find it very challenging, and therefore you might not feel comfortable with it. And the challenge is the simple question कि कौन सा command run करना है और कैसे run करना है. Okay, so what we hope with this course is to make you overcome that discomfort and that challenge, as to be able to say that you can use the Unix shell to wrangle files, which means move them around, transform them, change one file, reprocess them, and so on. Also to handle big data, like Mansi with 1,000 files, or I have Preeti, my own student, who has millions of files, not just thousands. Or manipulate spreadsheets. You may have a lot of spreadsheets. How do you manipulate them with each other? And to parallelize and automate your work. Okay, so these are the kinds of things we are hoping you're going to be able to do with this one. And do not worry too much about the nomenclature, but here is just a picture to show you what the Unix operating system is all about. So Unix is essentially a family of operating systems. It includes Linux and Mac OS, I've told you already. The terminal that I have been trying to make you familiar with, itni der se, is basically a program that allows you to interact with the computer using text-based commands. See, the important thing is the text-based commands. जो भी command हो, आपको type करना पड़ेगा वो. Click करने से नहीं होने का, right? And so this mechanism of texting instead of clicking is the command line interpreter usage. And that command line interpreter is the Unix shell. And that interpreter allows you to interact with the Unix-like operating system. And the Unix shell that we will be using for this course, also for the NIPGR Phyto cluster, which is the main thing for which we are doing this training for you, is the bash shell. And bash, therefore, you may consider bash as a programming language to work on the Unix shell. अब some of you may say कि अगर programming language है bash, तो फिर तो मैं R और Python भी तो use कर सकता हूँ. But that's not the same thing. ये जो people often interchangeably use terms like Unix, Linux, terminal, bash, command line, shell. Some people even say Ubuntu because इतना ज़्यादा Ubuntu use हो रहा है कि पूछो मत. But our Phyto cluster is not Ubuntu. Our Phyto cluster is, I will introduce the Phyto cluster to you very much in detail in the next course when you are done the basic intro Unix 101. The Phyto cluster is based on Red Hat Enterprise Linux. Okay. But a lot of free programs are on Ubuntu. And if you have programs on Ubuntu, they can easily be moved to the RHEL. So don't worry about not being able to use your programs from one kind of Linux operating system to another. Broadly, all of these words mean the same thing. So the idea is that this is a non-Windows, non-GUI, non-clicking command line interface. And the other reason why Bash, Bash is not like like an operating system is that Bash itself sits closer to the kernel or the hardware of your computer. The programming languages are absolutely on the outer side. So if you look at this like an electron shell in the picture that you're seeing on the screen right now, you have the hardware of your computer. And the hardware of the computer has a kernel around it. And that kernel has, just talking to the kernel at all points is the shell. And there are 2-3 different kinds of shells. And the shells are where you do, where you run basic commands such as listing, shell scripting, checking your directories, navigating file systems, remote logins, security, process killing, and lots of things that we will learn in this course. And the shell is very, very close to the kernel, therefore. And outside of the shell comes the applications such as R, applications such as Python, applications such as the C programming and the Fortran programming and the Java and object-oriented programming and the huge numbers of utilities. They are all part of the applications of your computer, of your Linux machine. Okay, so moving on. Yeah, I already told you that Bash is a programming language, but it is a little bit more special because it sits closer to the operating system. It is able to see all the files and folders in your computer, whereas R and Python and these other people are basically languages that exist in a very encapsulated kind of environment or an isolated kind of environment. So once again, you can think of Bash and the Unix command line generally as a way to orchestrate your analysis and launch other programs. Launching of R, launching of Python, launching of C++, launching of Java can all happen through the Bash interpreter. So even if you use some workflow management tools that are popular in bioinformatics, has anybody used workflow management tools such as SnakeMake or Nextflow? If you have used SnakeMake or if you have used Nextflow, they still have to be launched from the command line interpreter, from the Bash. So therefore, that is why the Bash is most important. And so the Unix command line is the primary way to interact with a computer in a scientific kind of computing context. And so therefore, you have to be able to learn why to do it. Okay. Another couple of reasons why you should learn the Unix command line interpreter is that of course, all the HPCs in the world use it, but also a lot of bioinformatics software that you might be using in your lab or others might be using in your lab and you want to learn is also written in Unix. Also, it allows very complex kinds of file manipulations, and we will see some examples just now. And it can be, and you can automate and reproduce everything. Like today, I clicked 7 or 8 different links or buttons, and I did this, that, and got my output somehow. If I have to do the same job tomorrow, I will have to remember, or I will have some trouble remembering what buttons did I click to get that final outcome. But you don't have to do that in Unix, because in Unix, you are writing the scripts down and you can just repeat the exact same text, what you wrote last time. And if you've forgotten what you wrote last time, there is another command called history. History tells you what you wrote when you logged in last time. Okay, So there's a lot of fun that we are going to have on learning the Unix system. So here's another motivating example for you. Imagine that you have several hundreds of DNA sequences, and all your DNA sequences are stored in collections of FASTA files. Does anybody know what's a FASTA format? I know many of you know the FASTA format, so I'm not going to ask that question. Don't put a yes. I'm assuming all of you have seen the FASTA file, but those of you who are not familiar with the FASTA is a text-based format where each sequence, as you can see on the screen right now, it is represented by a name. And it always starts with the greater than character, followed by a sequence in the next line. Okay, so as you can see, every FASTA sequence begins with exactly the same identical character called the greater than sign. After the greater than sign, there is some series of characters, numbers, letters, or something. These are called headers. This is called the header of the FASTA file. And on the next line, the actual DNA sequence or the protein sequence or the RNA sequence, it is all written on the next line. So this is a FASTA format, and let us say you have lots and lots of FASTA files. Now can somebody tell me how many FASTA sequences are there on the picture that I have put on the slide right now? If each FASTA sequence is represented by one greater than sign, then how many FASTA sequences are there on this picture? And can somebody tell me which organism these FASTA sequences are coming from? Again, the information that I'm asking you, usually such information in a FASTA file can be got from the header line itself. Every FASTA file has a header line which tells you ye kiska sequence hai, kahan se hai. Also, there are unique reference IDs which will also be in the header of the FASTA file. So I can see all of you already seem to know the exact meaning of a FASTA file. But if anybody does not understand or did not understand, then the first line, look at the first line. I've made two red circles basically to show you that there are two FASTA sequences on this picture. And both of them are human sequences because जो रेड सर्कल्स है उसके थोड़ा सा आगे आप हेडर लाइन को देखोगे तो वहां पे लिखा हुआ है होमो सेपियंस क्रोमोसोम्स. There are two chromosomes, chromosome 1 and chromosome 17. So these are the two pieces of, pieces of DNA that have been taken out from these two different chromosomes of the human genome. Okay, so let us say now you have hundreds of DNA sequences. यहाँ तो दोनों मैंने एक साथ लिख दिए हैं, but suppose आपके folder में 1000 files or 1000 files कहीं पे हैं. So hundreds of files. And now the question is, in hundreds of files को मेरा क्वेश्चन आपसे है कि कितने फाइल्स हैं. तो क्या आप अपना फोल्डर खोल के एक एक को काउंट करना शुरू करोगे? How are you going to tell me how many sequences are there in all in your entire folder? सारे के सारे फाइल्स मैंने बता दिया आपको, hundreds हैं और एक जगह पे रखे हुए हैं. अब उन सब फाइल्स को आपने ये मुझे बताना है कि कितने सीक्वेंसेस हैं, और आपकी सुपरवाइजर ने पूछा है how many sequences are there. So from a GUI, if you had a GUI, then you would have to go down, and there's no serial number on GUIs. When I showed you the GUI system, there's no serial number. There are either you can put a list or you can put icons, but there is no way to count that, right? But in Unix and in the Unix shell, there are many, many ways of counting how many files are there. So now Subhashish has put an answer that you can count the lines. But Subhashish, if you counted each line, do you see in front of you this file that I pasted, the two FASTA sequences? If you counted the lines, you would count, there is 1, 2, 3, 4, 5, 6, 7, 8, 9, 11 lines on this. But 11 lines is not the number of FASTA sequences, right? So the number of sequences, there are just 2 sequences, but the number of lines are basically 11 lines. So that is not the correct answer, Subhashish. So we have to think of a way of counting how many sequences I have in total. All of you just looked at this picture and you answered the correct answer that there are 2 sequences, even though there are 10-15 lines. It was because you were not counting all the lines. You were counting something very, very specific, right? What were you counting? You were not counting lines. You were counting, yes, Rajarshi, absolutely right. You were counting by whatever mechanism, you were basically counting the unique character that is signifying the beginning of a FASTA sequence. So we have to learn, Swapnil is absolutely right. Essentially, we have to find out through by wrapping our heads around Unix that we have to know how many files are there. So essentially, if you are now, let us come back to this task. You have a folder. That folder has hundreds and hundreds of. अरे मेरा screen share नहीं हो रखा क्या? मेरा screen तो share हो रखा है. क्या बात कर रहे हो? Have I not shared my screen? And nobody tells me. और इतनी देर से मैं लगी हुई हूँ बात करने के लिए. Really? I'm in the meeting. What do you want to share? No, my screen is already being shared, I think. Okay, let's see. I'm glad. Yeah, you guys have to tell me if something happens and you are not able to see my screen. Are you able to see my screen now? Oh, this is very weird. So it is not visible now. Okay. Kab ruka? I mean, let's, once you start to see the screen, I will ask you कि कहाँ पे हम रुके थे और कौन सा slide आपने miss कर दिया था. Of course, all of you were answering questions about the FASTA. That means you must have seen the motivating example. Okay. Thank you, guys. So now I have this second device login only to be able to tell me such issues. So I can see my own screen on the other device now. Very good. So I'm going to assume now that all of you can also see. Okay. So this was the example I had wanted to show all of you. Okay. And in this example, so where's the question? The question I'm asking is, imagine that we all have hundreds of files. Imagine that you have hundreds of these FASTA files and they are not sticking together in one place like you are seeing on the screen. On the screen, it is easy for me to chipka two sequences and to show you and ask you kitne sequences hai. But actually in your folder, imagine you have hundreds of little icons and each icon is a file. And inside each file is this header line and the sequence, right? And the question is how many sequences? So let us divide this question and break this problem into 3 steps. And this is how you will think when you are thinking as an individual who's working on Unix and trying to chop up your task. So the first job you have to do is, I don't know how many files there are, but I'll combine them all. And I also don't know whether each file has one sequence or two sequences or more sequence. So I'm going to combine all the files together and then it will all become one file, just like the way I've shown you on the screen. On the screen, there is a single view where you see two files, two FASTA sequences. So we are going to first combine all the files, however many they might be, into a single file. And then we are going to search for any lines that begin with the greater than sign, right? And once you have found the lines which begin with the greater than sign, gosh, okay, hold on, I gotta pick up this call now. Okay, can everybody hear me now? I'm back in the meeting. Saurav, Renu, are you able to hear me?
Yes, ma'am.
Okay, great, thanks. So, so we are going to break the problem into small pieces. First, combine all the files and then search for that greater than sign and finally count the number of lines of text that have the greater than sign, right? And this, all these things, these three different things are not three different tasks. These are basically, they can be done in one single Unix command. So imagine like this. You have you have a FASTA file, which is here. It has 3 sequences. You have another file, which is here. It has another 3 sequences. So we combine. So like this, there are hundreds of files. So if we combine these 2 files, you get a single file. Do you see the single file and how many sequences does it have? 1, 2, and all the way to 6. And then you pull out only the greater than sign. So search for the lines that start with the greater than. And see, there you got the lines with the greater than. The moment you get the lines with the greater than, now what you do is what one of you told me earlier, कि straight away count the number of lines. इन lines को count कर सकते हैं और इनको count करते ही आपको पता चलेगा that these are 6. So that is how you find the total number of FASTA sequences in an original set of files, which could be each file with many FASTA sequences or each file with a single FASTA sequence and so on. And what did we do here? We did 3 steps. The first step was, what was the first step? Combining the 2 files. Second step was what? The second step was search for the line that starts with a greater than. And the last step was simply count the number of lines that you get in the output. So all of these 3 tasks are run with this Unix command. And all of these 3 tasks can also be changed together into a single. So the first task is basically combining this and this. And the combining of this and this happens with a cat. So cat is a concatenate command. I will talk to you and teach you the concatenate command later. But right now it is just for you to know that you can cat 2 files or 3 files or hundreds of files by saying cat all the files star dot fa or FASTA. And everything gets combined together into something like this. And once you have this, you want to just search for the lines that have a greater than sign. Now, what is the command that allows you to search for the lines that start with a greater than sign? There are many different ways to do this, first of all. Okay. All the roads lead to Rome. Sometimes it's true for the Unix command because there is no single correct answer. I feel that the correct answer is always the one which is fastest or shortest. But if you feel more comfortable with the lamba answer, then you can use that also. So then you perform the second step, which is search for the greater than sign. How do you search? You search by using one way of searching is to use the grep command. The grep, grep is like grabbing something. So grab everything that has a greater than sign. And we do that exactly right now. We grab everything that has a greater than sign. And the moment you grab the greater than sign, all the greater than signs come into one file. And once all of the greater than signs are there, you simply do a count. And what is the command that does counting in Unix? I will teach you that by and by, but it's called the WC command. So WC minus list. So count and then list the number of lines. So when you say WC minus L means you're giving an argument to the command, which is about word count. WC means word count. So you're word counting, but you're counting only the length of those words that you found containing the greater than. Okay. And the length is a single number. And therefore your final answer is number 6. So if I asked you a question, how many files were there in your folder? You could have simply written a single command, which is basically chaining together 3 different steps. And each step in itself is a very simple step. Chaining them together is also a very, very simple step. And you do that and you'll get the answer 6. Is there anything here that anybody did not understand? I don't need you to remember or learn or ratto, what are these commands? That is all about later. But did you understand the breakup of how we broke the task and made it 3 steps? And then how we combined the 3 steps into a single place and chained these commands together and got the final answer. This reason or this example has been shown to you solely for the reason that you should be able to run this on your own at some point or think in the same way on your own. Okay. So what we are going to do now is, where do we begin? Okay. All right. This is a motivating example for you. In this course, all right, let's come to this course. And in this course, I'm going to teach you very simple stuff. Mainly the things that I want to teach you are the fact that introduce you for the first time to Unix or Linux. Those of you who were not already familiar with Unix or Linux, have it here now, right? And that we've done. The second is the introduction to the command line interface, which also I believe I have done till now. The third is file and directory management. Now I have taught you about file and directory management to the extent that I told you how the files and directories look like in a Linux or a Unix environment. But now we will learn some basic commands for file and directory management, or we call them file navigation commands. And then we will try to go on to file permissions and we'll do some text editing. Now, I need you to understand that this is some of you, I'm going to, the main aim of this course is to teach those of you who have never used Unix before, those of you who have never used the command line before. And therefore, you have to tell me if I'm going too fast. Of course, some of you have used Unix and some of you are good with it. And therefore, some of you are able to answer a lot of questions that I'm telling. And that makes me feel कि सबको आता है. But it's very, very important. This course is not meant for people who already have learned Unix. It is mainly meant for those of you who have never used Unix. So please, I encourage you to speak up and ask. I've written that this course has A, B, C, D, E, F, G, but we will do absolutely nothing till you have understood every bit. So even if we cannot complete the course today, it is important that whatever we do complete, that much you should understand. If you don't understand, please stop me, ask me. And I see Rakesh has a question. I'm very glad that there is a question. Please feel free to ask these questions. So the point I'm saying is that it is not important to complete the course. We can meet again and again. I'm right here in the institute and we will always take these 2-hour lectures, which will hopefully allow you to understand something better. So today we are meeting from 3 to 5. I'm going to try ki Wednesday ko bhi we can meet from 3 to 5. If some of you are interested, we will continue this course between 3 to 5 p.m. on Wednesday also, if that is okay for all of you. Okay. And so the question that Rakesh is asking is, if two FASTA files are in two different folders, then how do I combine them? Because if it is the same directory, that is much easier, but sometimes they are sitting in different folders. So Rakesh, that is not a problem at all. Once you learn how to navigate the file and directory structure, let us say you have a file structure where you have your directory called data shell. And within that data shell, I just showed you there were two directories. Let me show you पीछे वाले slide पे कहाँ था. हमारा layout कहाँ पे है जो मैंने आपको एक basic example layout का दिखाया था, ये वाला, right? So here, do you know what is your main directory's name? The main directory under which you have 3 different directories and 2 different files. Does anybody know what is the name of that folder? You can look at the GUI if you find the text-based system difficult, but the answer is written in both the pictures now. Who's going to tell me the name of the folder? The main folder under which you have 3 folders called coronavirus, molecules, sequencing. So what is that main folder's name? Is it your desktop? Is it your downloads? Is it the documents? Or is it somebody else? Oh, come on, I'm looking at the chat box. Yes, so Baljinder says it is the data shell. Ashish says and Radhika says it is the data shell. But I find that again and again, Rajarshi and Baljinder and Ashish are answering. Is there anybody else who wants to answer also? I want to encourage you guys to, interact with this course. If you don't interact, apne se sometimes easy nahi hota learn karna, right? So thank you, Diksha. That is a new name. I'm glad. So this is the data shell. And Subhashish has an even better answer ki data shell desktop mein hai. Very good, Subhashish. And Shubham, you're absolutely right. So coming back to the question that Rakesh was asking. Now, suppose Rakesh has 10 FASTA files inside molecules and he has 10 FASTA files inside coronavirus. And I'm asking him ki kitne hain, jaldi batao, jaldi batao. I am his supervisor and I'm like always carrying a gun on me and I'm always disturbing and asking, batao, jawab do. So if Rakesh wants the answer quickly to how many FASTA files are inside coronavirus and how many FASTA files are inside molecules, so he can combine both and find the answer separately also, together also. So separately, though, I just taught you how you go into coronavirus and perform that single command line where you chain up 3 commands, you break it into 3 steps and get the answer. Same thing, you go into molecules and do the same thing, but that is also repetition. Why should you go into molecules separately and into coronavirus separately and into sequencing separately? So a good way is to be able to do it from the data shell. The data shell is the parent directory of these 3 directories, right? So now you're sitting inside the data shell. So you come back upstairs, one step up, come out of coronavirus into the data shell where you are. Now, data shell, you can simply, instead of saying cat star dot fasta, you say cat star slash star dot fasta. That means star dot fasta या बाहर रखा हो या इनमें से किसी भी डायरेक्टरी के अंदर हो, उन सबको एक साथ cat कर दो. So it will cat everything together. Okay. So we will resolve this by and by when we come to that point. But to answer your question, Rakesh, this is a very simple task and it can easily be done. And I'm very glad that you are coming up with questions like this, which means that you are thinking कि मैं इसको कैसे address कर सकता हूँ. Okay. So जहाँ पे यहाँ पे first command देख रहे हो, cat star.fa. That means all the FASTA files which have suffix called fa are the FASTA files. All of them get concatenated. And I'm writing star.fa. That means शुरू में चाहे जो भी नाम हो, file 1 हो, file 2 हो, file 10 हो, file x हो, star.fa में सब cover हो जाएगा. And the star means कि इसी directory में. But if you put a slash over there, then it will mean कि इसी directory में नहीं, बाकी इसके अंदर जितने भी और directories हैं उन सबको भी check कर लो और वहां से भी FASTA निकाल लाओ. Okay, so you can do that. So Mustafa says cat slash dot FASTA into merge FASTA for something that is in different folders. So does cat $1? So okay, now Mustafa has already written a code for you. Mustafa, don't thank you very much. I think I will get you to meet Rakesh and you can solve this for him. But yes, this is how we need to help each other and be able to sort this out. So Mustafa has given one way. There are many ways in the same way to answer the question that you asked, Rakesh, but it can all be done in a single line of code, exactly the way we are telling you. Okay, so coming back to this course, we are going to do file permissions, text editing, input, output, redirection, and pipes. जो अभी मैं आपको बता रही थी, ये pipes and redirection है. Searching and filtering, जैसे कि मैंने अभी बताया. Second step में searching करा हमने. You should know how to do searching. Also process management. प्रोसेस मैनेजमेंट में ये होता है कि कई बार आप गलती से कोई प्रोसेस स्टार्ट कर देते हो और उसका कोई अंत ही नहीं है, उसमें कोई रैंडम लूप लग गया. So you need to be able to kill it also, especially when you're working in a high performance computing environment where other 20 people may be working with you. You cannot take up all the space. And maybe sometimes you start a long code, it starts running, you made a mistake somewhere. And if you made a mistake, somewhere, then you cannot, you should not run that code. And so a good idea would be to be able to be able to kill that process. So you have to learn process management. That is my point. Okay. Then shell scripting fundamentals. So what Mustafa just typed in the chat box was a shell script. So you have to learn some basics of shell scripting. And then of course, more often than not, what we need is help. And I am here today with you. Mustafa is here online. Some of the other people are here to help you, but we are not there all the time. And therefore you need to know how to access help and how to access documentation. Okay, so that is also going to be covered. Now, moving on. Right, navigating files and folders. So let's say this is how your Linux, a general Linux computer will have something like this kind of a directory structure. So there is a root. Every user will have a home directory, and that home directory will generally contain all the users. So in this home directory, there are 3 different users. One is the Ubuntu user, another is the GYLab user. And for the NIPGR Phyto cluster, I've specially written GYLab here so that you all know that for each of your labs, a home directory has already been created on the NIPGR Phyto cluster. If you have already been able to access the Phyto storage, the Phyto Vault that we have, which we call B, then you would know that your lab is called something like GYLab 204. So that's my lab. And all of the researchers in my lab can access that common area, which is our home directory. In the same way here, there are 3 users, Imhotep, GYLab, and Ubuntu. And within each of these users, their home directory, they have several other things. So every user like GYLab will have my own desktop, my own downloads, my own documents, just like you do on your Windows computers and elsewhere. Ubuntu also has a desktop, documents, downloads, etc. Lots and lots of things. And in the desktop, all of you just saw that we have a data shell. And in the data shell, how many directories? In the data shell on the desktop, how many directories? I'm looking at the chat box. If anybody remembers, iske niche kya hai? Very good, Ritama. Thank you. Radhika, you're absolutely right. There are 3 more directories within this data shell, and we don't know how many more. Coronavirus के अंदर क्या पता और हो, पुराना corona, नया corona, sequencing में बहुत कुछ हो, हर जगह बहुत कुछ. So the directory structure can be very vast and very much like a maze. But if you know how the order goes, then if you know the path, then you can navigate from one folder to another very easily. So the first rule of law in navigating files and folders on the Unix shell is that you have to know that you have your home directory. Okay. Now, if I am GY, my home directory is the GY lab here. If you are from Shailesh's lab, then your home directory is SK lab. If you're from Alok's lab, then your home directory is AK lab, and so on, right? Now, the location of the files and directories in Unix is always specified with a path. So tell me the path of the data shell now. The relative path of the data shell is from your home directory. From your home directory, if you want to go to the data shell, whether you are Ubuntu, whether you are GY, whether you are Imhotep, or whether you are any other user, the path to the data shell from the moment you log in, when you log into a server, you come straight to your home directory. From your home directory, if you want to go to data shell, you have to go to desktop and data shell. That is the relative path. So the path can be relative to your home directory, but there is also an absolute path. Absolute means absolute truth. That means that path, regardless of whose user you're in, you can straight away go to that directory. So the absolute path for the data shell directory, which I've colored on this page, is the yellow path, starting from the absolute top, which is the root. And the absolute path always begins with the root, which means the root sign slash, which is the top. Okay, the root or start of the file system is like the door to a building, which I've shown on the right side in a little picture. So there's a door to the building, which is your root. And from the root, you can go to the first room, the second room, the top floor, the kitchen, the bath, the study, wherever you want to go, the balcony, and so on. And this is an important thing to remember, because if you are sitting in the desktop and if you're looking at the data shell and you're searching for a file that you downloaded means you downloaded it from your email. Usually the email downloaded files go into which directory? The downloaded files go into downloads or sometimes into documents. But if you're sitting in the data shell and you're thinking that my download is missing, obviously it is missing because the download is sitting in another path, in another directory. So you have to have a grasp of the directory structure. All of us who have been using Windows and computers and laptops for years and years, we all know the whole concept of the path. अगर हम डाउनलोड्स में हैं तो हम डेस्कटॉप पे नहीं सर्च करते, अगर हम डेस्कटॉप पे हैं तो हम डाउनलोड्स में नहीं सर्च करते, राइट? हमें पता है कि हम डेस्कटॉप पे हैं इस वक्त. In the same way on Unix also, somehow I feel that students are not able to understand the directory structure or navigate the files and folders in a Unix environment, even though you are already doing it on Windows. So you have to just close your eyes and think about where you are and build a map of the whole directory structure around you. And then you will know where you are. So let us say now we are in the home directory. You have a home directory. You are Ubuntu and you have the home directory and you want to go to documents. So what is going to be the relative path to get into documents directory in your folder? Can anybody type in the chat box and tell me? You are the Ubuntu user and you need to go to documents. How are you going to go to documents sitting in the login space of the Ubuntu user? So Sankadeep says that I will type home. You don't have to type home and Ubuntu because you are the Ubuntu user and it's the relative path that I'm asking. That means you are already in home and Ubuntu and you are already, so you don't have to go to downloads. Yeah, you simply say CD to document. So you just go to documents. That is where you are. If you are a user and relatively you have to go. Okay. So this is the difference between absolute and relative path. Absolutely correct, Ruby. If you are the Ubuntu user and you want to go to documents, you just say change the directory to documents. And why did you type CD? Everybody is giving the answer as CD. CD is meaning change to the directory. But I'm not asking you about the command. I'm just asking you the path. The path would be simply documents. Okay. So we go there. Now the question arises, okay, now you know the path. How do you go there? So if you want to go somewhere from somewhere, you have to know how to navigate. And this navigation has certain commands which are very, very, very important. So let us begin the all right, let us first see how your terminal looks. So if you're on Mac, how many of you are on, why don't you do one thing? Type in the chat box, what is your operating system? I need to have some idea about how many of you are accessing this meeting or this course from a Mac or a Windows or a Unix machine. So I see 2, 3 Macs and I see 2, 3 Windows. Is anybody accessing this? Okay, again Mac, again Windows. All right, 1 or 2, only one Unix till now. Second, Janvi is also on Unix. Kanka Mukherjee is on Linux. Very good, Kanka. Brishti is accessing from Windows. So Brishti, are you having a terminal on your Windows machine? Diksha has got a Linux. Kamakshi has got a Windows. So Kamakshi and Brishti, my question is to you. So how did you access the terminal? Did you get a putty? Are you using the putty or have you got some other app which is letting you view the terminal? Like a Unix environment on your Windows machine? And this question is for those of you who are using Windows devices to log into today's meeting. You can type your answer in the, okay, Brishti is using WSL. Kamakshi, what are you using? Do you have, Rajarshi is also using WSL for Windows. Very good. So is Rishabh. Kamakshi, what are you doing? So see, if many of you are preferring WSL, then what I'm going to do is I will make sure that next time we help others who are using Windows and they have never been able to connect to the terminal, we can tell them कि भाई WSL ही ले लो, सब लोग के पास वही तो MOBA exterm कोई नहीं use कर रहा. हम लोग ने email में MOBA exterm भेजा हुआ था कि ये try कर लेना. See, but we ourselves are Linux people and we ourselves are Mac people, so we don't usually know what Windows users prefer. I'm very glad to see this answer from all of you. So you guys have, so how do your terminals look? See, the Mac terminal is very colorless and boring, just same background. Best thing you can do is pura background ka color change kar sakte ho. But look at the Ubuntu. In the Ubuntu, the terminal is so cool. It's got colors for directories. It's got colors for the name of the computer. It's got colors for command lines and so on. So इसमें से आपका terminal कैसा दिखता है? Colorful या bland, plain? That is what I want to know. Okay, very nice. So Rajeshi says that the Windows people have very similar to Ubuntu, colorful, kind of easy to understand and wrap your head terminals. Very good. And others, Swapnil and others still have plain, just like myself. So what I did, Swapnil, I had a plain, I had a plain terminal till I decided to not use my terminal, but I used item. So on the Mac, I like to download the item, and the item has some colors which makes it interesting. And you can fool around with item layout and visuals, and you can, that is quite interesting. So try that if you can. And Mac also, Sankadeep says Mac may there are other ways. So even I'm using Mac, and so it's good if you have some colors because it keeps you awake a little bit longer. That's all. Okay, so let's get started, everybody. So now you're going to write your first command. अब तक जो भी आपने लिखा समझ आया कि नहीं आया, forget about it. I want you to run your first command, which is like the hello world business when you learn something new. So I want you to type, type out something that will make your computer or your terminal say hello to you. So you're going to type echo hello Unix. So echo is a command, of course. The reason you've typed echo is that echo is a command. Echo means you get back the echo. Whatever you say comes back at you. So echo, hello Unix or hello world. Type that out and I'm going to do it too. Let's do it together. I'm in the meeting. Here's my terminal. So, इसका size थोड़ा सा बड़ा करते हैं. Shashi will tell me how to make it big. बड़ा हो गया. Can everybody see what I'm typing? I'm sure you can. थोड़ा सा बड़ा screen मैंने करा है. And I'm going to echo. I'm going to get started. I'm going to say echo hello world and put it in quote unquote. And then what you get is the hello world being echoed back at you. Try something else. Echo hello Unix. Okay, and those of you who have never used Unix before, I want you to understand the value of this. You have actually used a command just now to interact with the computer and make the computer say something to you or print something for you, right? So it is echoing a hello Unix this. Now, if you want to ask the computer, आज की date क्या है? घड़ी में देखने की जरूरत नहीं है, फोन पे देखने की जरूरत नहीं है, सीधा अपने टर्मिनल से पूछो what is the date. Don't put a question mark, just write date and let's see what comes. So in my case, right now I'm in the hello, I'm in the EDT. EDT mein kahan se? I should have been in Indian Standard Time. So it is September 8 today. And it's very early morning, 6:53 a.m. on the eastern side of the United States, which I don't think is what we need to know. What we need right now is the Indian Standard Time. So we need to fix this. Shashi, this is something that on the training portal, we have to change the time zone to the India time zone. This is a good thing that I've made notice just now. So let's fix this. And on your terminals, now all of you are using terminals. Not everybody is using the Phyto Learn Abhyas. I want you to tell me if you are all on the EDT time zone or is anybody on the India time zone? Because on my laptop, which is here, which is my Mac laptop, when I use the iTerm, there I know it's 16:24 Indian Standard Time for me. So those of you who have never used Unix before may have a unique time, but if your machines are fixed on the correct time zone, then this will tell you. So Shashi, agar aap hain, to yeah, Saurav, please make a note that maybe the Phyto Learn Abhyas server is not on the Indian time zone. So we have to fix that currently. And that is, I think for you, it will be, for us, it's very easy to fix it. But good. So this is the date. What else? Echo something else to yourself. That is what DIY. Echo something else to yourself. I'm going to echo. Can I make the, why don't I ask this guy to say hello to me only? Hello, GY. So that's it. So the machine is saying hello to me rather than to Unix or to the world. Okay. So try this out yourself and let's go right back. You can echo stars. You can echo just about anything that comes to your mind. Okay. Is there anyone who has not understood what is the meaning of the echo command? So we gave two commands here in this DIY example. The first command was an echo command and you made something happen. The second command was the date command and you got the date of the current date, not just the date, you also got the time. Remember? No, it didn't just tell you. So what does the date command tell you or return for you? The date command tells you, first of all, what day of the week this is. And then it tells you what month of the year it is. And then, of course, it tells you the date. And then it tells you the exact time to the second at which you asked or you gave the command to the computer. And then it tells you what time zone you're in. And finally, it tells you what year it is. Okay. So that is the output of the date command. Is there any confusion that anybody has? Any questions that you have till now? Or can we move on? Because now we are going to get started and we are going to see what these different commands do. One important thing I want some of you to understand who are using Unix for the first time is that commands are very easy to recognize. They may not be easy to remember, but they are easy to recognize. Okay, so echo basically means you're asking the computer to say it back to you. Date is date. Just now we were looking at ls, which is listing. Somebody else said during the directory path slide that you can use a cd, which means change the directory. So And I was showing you in that quick motivating example that there is a command called WC minus L. So basically that is a word count command, right? So if you think about this, the commands, the simple basic commands in Unix are so easy. Either you can recognize just by reading, or else you know that they are the acronyms for something even easier. Okay, so Unix is very, very easy, and it will, but everything, like with everything in life, practice alone is going to make you perfect. So these are some of the basic file navigation commands, and I want you to try these. Very good, Rajarshi. Yes, I wanted somebody to notice that, and I'm glad that you have noticed it. So great. So moving on, basic file navigation commands are given over here. There are many, many more also. Can some of you type the history command and tell me what you get? If you type the command history, what do you get? It basically should list all of your previous commands, whatever you gave just now with me. And some of you can tell me, okay, my history is 5 steps long, my history, and there will be a number to the history. Basically, history tells you a maximum of certain, I think 400 to 500 commands it will tell you that you did in the last, whenever, the last 500 commands or 400 commands that were written. But agar aapne sirf do hi commands diye ab tak, to your history will return to you 2 lines. So I want to know how many, first of all, type history. Let's do it together. But now, how much, what is there in your history? How many lines of code do you get in your history, when you type history? And you can type in the chat box what is the result of your history command. And I will also do it in front of you on the Phyto portal, Phyto Learn portal. History. Okay, I've given 6 commands to my history, and Ashish has done 5. And so very good. So all of us have, Ruby has tried more. Radhika has not been doing a lot of work, but Kamakshi is doing more than what anybody else is doing. Very nice. Thank you, guys. So at least 4 or 5 commands we were looking at. And there, of course, the moment I know Baljinder's and Rishabh's number, I know that they've got countless. So I am very surprised, because Baljinder and Rishabh, I always thought that there is a limit to history, that you can get a certain number like 400, 500. But I can see Nitish has commands going back to 2000. So it is possible that you've not put any limits on your history, or that history continues forever in the past. Very, very interesting. Okay, great. Wonderful. Brishti also has 500, so things are good. So we have a lot of students who have used Unix before, and those of you who have 3, 4, 8 single digit or even double digit history, I'm glad I asked this question, because ab hi se main answers sare puchungi. So now, Shubham, you and Radhika and Ritama and Ashish are in trouble, because now I'm going to, and Ruby, I'm going to expect the answers coming from Kamakshi and so on. Okay, so this is a good one. It has told me which of you are not to be coaxed to get an answer from. Very good. Carrying on, carrying on. So we are going to move on, and I want you to type something else also. What is your present working directory? Do you see? If you want to know your present working directory, whether you are in documents, or you're sitting in desktop, or you're in the data shell, or you're in the coronavirus, you have to type the command pwd. Pwd means P for present, W for working, D for directory. So the present working directory. Okay. So type that out, try it out, and tell me what happened. CD means changing the directory. You can change the directory to any space that you want by giving a path. The path could be an absolute path, or the path could be a relative path. So CD to something else. But if you type CD, okay, Ritama is in this one, Rishabh is somewhere, Shankadeep is some, very good. So the question that I have to ask you guys is, if you wanted to go one step backwards, upstairs towards the root, then you will type dot dot, cd dot dot. Okay, and when you want to go forwards into a new directory, then you're going to say cd dot slash the name of that directory. Okay, but you don't have to remember it. We will learn it by and by. We will try it ourselves together. So who's going to tell me the difference between ls minus L and ls minus A? Why don't you do one thing? You type ls minus L and then ls minus A, and tell me, how did you feel? Both of these are listing commands. So when you do an ls, it will give you a list of commands. The present working directory, all of us have done together just now. List also all of you have done. Maybe there is nothing in your directory, so you will find nothing there. But if you put ls minus l and ls minus a, then what happens? And cd/tmp will probably take you to not the temp directory in your home, but the temp directory in the root, because you've taken it straight to the root. So don't worry about the last two commands. I think that is too much information for the first timers. Why don't you just try ls minus l and ls minus a? I will also try it with you and let's see what we get. Because these files are empty right now, ls gives me, okay, there is r. ls minus l, okay, ls minus l tells me that there is a directory in my this one. And when I do ls minus a, aha, I get much more. How about ls minus LA? There, ls minus LA is better. Can somebody tell me what is the difference between ls minus ls minus LA and ls minus just L? When I did ls minus L, I got a long list of the directories that are visible in my area. When I did ls minus LA, added an A flag to it. That means all the files and all the directories, even the hidden ones. So you find that the ls minus LA showed me a lot of hidden directories where I have, of course, the bash RC and the bash profile. When you log into the portal, you see something, you see messages, and the fact that you're in bash is also within the hidden files. So this is where you get a full listing of all your files if you do an ls minus LA. Now again, some of you may be wondering, but nobody has asked me, ls is a listing command. What is the meaning of making a space and then minus L and then a space and then minus LA? What is this whole business of minus something? A command is a command, whether it is echo or it is date or it is cd or it is ls. Why are we putting a space and then minus L, minus LA, minus H? Can someone tell me? Or is that a question in your mind also? If so, then I think I have to teach you the basic command structure. Okay. And the basic command structure is given in the next slide. So here is a basic command structure. It is the overview of the general structure of any command in Unix. So the command itself will just be a single connected word or series of letters such as ls or cd or echo or date or wc, something. That is the command, right? Uske baad we put some flags such as the one I just told you. And these flags always have to come with a minus, okay? A minus. So the minus here is minus L, ls minus L, which means you're doing the listing, but you can put it with a minus L to get a long list. Or you could put it with a minus LA to get all the listings of the hidden files also. But the minus itself, the flag itself, the minus L or minus A or minus H or minus whatever, it has no value. However, sometimes you have to do a value. You have to add a value to the flag. And if you put a flag with a value, then it is always used with a double minus, not with a single minus. A single minus flag means an argument with a no value. But if you put a double minus, such as ls minus L minus minus sort, then you have to add a value. Sort करना है आपको जो ls में listing आएगा, suppose hundreds of files are there, उनको sort करना है. Sort किस basis पे करोगे? On the basis of which value? So you have to give a value. Suppose you want to sort all of your files based on सबसे पुराना कौन सा है, नया वाला कौन सा है, latest कौन सा बनाया है. Then you have to do ls minus LT. Basically, ls minus LT lists everything with the time. But ls minus L minus minus sort time gives you those files by sorting them into time. Okay. And then sometimes you also have positional arguments. Positional arguments means कि find me the listing of not just the current directory, but find me the listing of the directory which is the documents directory. And the documents directory is पीछे, आगे नहीं है, नीचे नहीं है, ऊपर है कहीं पे, घर पे, है ना? और वो उसको उस argument को कहते हैं positional argument. So this is something I need you to understand right now. Sometimes you will find commands that have a minus with them. Some commands have a double minus with them. Some commands don't have a minus or a double minus, but a tilde there. So that tilde here is a positional argument because that is a path. Okay. So how do you know? The next question obviously is कि आपने तो ls minus l बता दिया, ls minus lt बता दिया, ls minus lh बता दिया, ls minus la बता दिया. How do I know और कितने ls minus something है? Does anybody know how? And that is how you access help. So if you want to access help for any command, then you have to just give a flag with an h argument and no value. So अगर ls की help चाहिए, listing की help की, I'm just working alone. Now let's say I tell आशीष फटाफट अपने पूरी जो डायरेक्टरी आपकी पीडीबी फाइल्स की है या ऋषभ के पास थी, किसके पास वो डायरेक्टरी थी पीडीबी फाइल्स की, पीडीबी फाइल्स में से मुझे बताओ सबसे बड़ा पीडीबी फाइल कौन सा है. तो उसको सॉर्टिंग करना है और उसके साइज पे सॉर्टिंग करना है, टाइम पे नहीं सॉर्टिंग करना. तो आपको क्या करना होगा, आपको कौन सा ls यूज करना है, कौन सा फ्लैग यूज करना है, उसमें क्या आर्ग्यूमेंट डालना है, क्या वैल्यू डालना है, ये पता ही नहीं है. And supervisor ने कह दिया कि बताना पड़ेगा. So one thing is you go and ask your friend who's doing Unix, and of course you can come to our lab, and all the students in my lab are more than happy to help you. But it would be good if you knew the answer yourself, or if you could find the answer yourself, or if you knew where to find the answer. So Unix is a wonderful operating system in the sense that it always tells you कि help कहाँ पे मिलेगा. Okay? और help यहाँ पे कहाँ मिलेगा? H में, H flag में, H meaning help. ls minus H करके बताओ क्या आता है. Help लेते हैं. Let me go back and try this myself, or I would like to get some help on the cd command also. ठीक है? So ls minus H try करें. कुछ भी नहीं आया. Minus H. Oh, minus help. That's it. So what you want is minus help. Okay. अब ये हेल्प जाते ही पता चला देयर इज अ वेरी लॉन्ग हेल्प, बहुत ही ऊंचा, बहुत सारा. तो ls माइनस हेल्प में बहुत लंबा आंसर है, तो लेट अस टाइप इट इनटू मोर. धीरे धीरे पढ़ते हैं, मोर नहीं, लेस, धीरे धीरे पढ़ो. सो दिस इज इट. The usage of the ls command shows that you put ls and then list the information about the files in your current directory by default. If you put ls minus a, it gives you all the information, right? So it does not ignore any hidden files and shows you everything. But if you put ls minus capital A, it gives you almost all files. Very interesting. ये मैं भी भूल गई थी. ls minus b is basically, it is printing out a C style escapes for non-graphic characters. ls minus c is something else. धीरे धीरे नीचे जाते हैं. ls minus d से डायरेक्टरी पता चलती, केवल डायरेक्टरीज पता करनी है तो ls minus d करो. ls minus f का भी कोई मीनिंग है, ls minus g का भी कोई मीनिंग है. चलते चलो. Look at the ls options. You can print so much here. Okay, kiki bytes kibi bytes में 1024 bytes में you can find the size of the listed files. Okay, indicator, hide control characters, recursive ls, which is ls minus r. Very good. All right. And sorting कर सकते हैं. Minus sort करके आप उसको word से sort कर सकते हो, extension से sort कर सकते हो, size से sort कर सकते हो, time से sort कर सकते हो. Very good. And then you can do ls minus w, which means the width, ls minus z, which is the context. The size argument is usually an integer and an optional unit. Do you get it? So ये सारा का सारा help जो है, ये आपके पास जब भी extra time हो और आप बहुत वेला feel करें अपने आप को, तो आप कृपया इसको पढ़ सकते हैं. But what I want you to know is that every single command in Unix has a lot of help. And you can access the help by doing this. So let us say I want date, and date may I want some help. What happens? Again, I want to read its detailed answer. So I want to read it, and I want to read it slowly. So I'm going to pipe it into less. And the usage of date is date universal date, or just in my current time. Date date does not have a minus a flag. It can have a minus d flag, minus f flag, also minus r flag. Okay. Anything else? Minus small r flag also is there. Very good. Anything else? Date you can set also. So there is a minus s there, minus universal date, coordinated universal time. Okay, ls minus u karte hain. Let's see. date minus U. What happened? There, just now I told you that when I do date, my computer gives me the time of the Eastern US scheme. But now I want to know the central universal time in the world, and that is Monday, September 8, 11 a.m. right now, as per the universal time calendar. Okay. What next? Similarly, how would you find? Now, I'm looking at the text box and I want an answer from some of you कि अगर आपको help चाहिए echo के लिए कि आप क्या echo कर सकते हो? तो how will you get the help on echo? Is that the same way? Echo space minus help or echo space minus h? Or is it something else? Try करो और मुझे बताओ. So if you say echo minus help, आपको लगता है इसमें help मिलेगा echo में? Yes or no? No, कोई help नहीं मिलेगा, उसमें सीधा echo हो जाएगा. Similarly, अगर minus minus help भी करोगे तब भी echo में help नहीं मिलेगा, वो echo हो जाएगा. So what is the other way of accessing the help on any command? There is one more way. Like I told you, there are many more ways actually, but I want you to learn one more way, and that is to study the manual of echo. Okay. Oh, Brishti has answered that already. Brishti, you're absolutely right. And I want, whom should I ask? Okay, I'm going to ask Kamakshi to run this. Kamakshi, try the manual on echo and tell me what you get. And you have to tell me whether you were able to find whether, tell me any one flag that I can use with echo. Just now I used the minus U flag with date, remember? So tell me one flag that I can use with echo. Can I use echo minus A? Or can I use echo minus B? Or I cannot use it. So you have to tell me, Kamakshi, what is the letter flag that I can use with echo? Ashish, tum batao. Either Ashish or Kamakshi have to tell me. You can type in the chat box. It's okay. You don't have to speak up. I also don't see any comments from Jagjit. Jagjit, are you in lab 102? Are you still with us? Are you able to understand any of this? Also, Samta. Is Samta here? Samta is at NII. So, Rajashri has a good question. Why echo returns help but not star? That's a good question. Find the answer, Rajashri, while I'm waiting for Avni and Kamakshi to tell me the echo ka help. Oh, you're not able to get the echo help on echo, Radhika, is that correct? No. Help echo. Yeah, there's the help on echo. So do you have a minus A or minus B here, or do we have something else? Let me check. We have a minus N, E. There is a minus A and B also. Echo interprets the following backslash escaped characters. No, there is no minus A, minus B. You can have echo backspace L. And Rajashri, I think that answers your question also. Backspace karke, you can probably get your stars to be this. ASCII codes, 8-bit characters. But I did get the answer from neither Kamakshi nor the students, nor Samta, nor Jagjit. Have we left you behind somewhere? Is there something that you're not understanding? Please feel free to type in the chat box or ask if you're not able to. What we are doing right now, the whole idea of this current slide right now is to make you understand the command line and the basic command structure. That commands are generally just one word, like echo or date. And those commands can sometimes have additional flags and additional arguments. And those arguments can have values, or the arguments can be positional and so on. Okay. Vijay has a command. How can we print any word with inverted commas with the echo command? That's a very good question. That's a very good question, Vijay. So what I would suggest is you put the inverted comma also within the echo command itself. Should we try it? Echo hello gives you a hello. Echo inverted quote unquote hello also gives you a hello. And the question he's asking is, ki, how about the quoted, this one also coming in. So let's try this one. The backslash usually is the solution to things, but that's not working here. Okay. So try this one. Now, okay, try again. Echo. All right. So we have a backslash and then a this one. And then we again have, let's try. Oh, again not found. Okay, let me try. I think this works on some shells. It works. You have a backslash. As long as your backslash is working, it works easily. And Rishabh has something else. Oh, echo minus E is also there. See, there are so many different ways. I don't know if it'll work here. Let's try. Echo minus E means exactly. No, Rishabh, it didn't work on this shell. So let's try echo minus E करके double character लेके backslash भी ले लो और फिर try करते हैं आया कि नहीं. Nope. Again, double character backslash single. And then done. I told you it's not impossible. So you add it again. Do you see what I'm doing? Now I've got it. So there is a backslash, but the backslash itself will not work. You have to hide the backslash, quoting it inside these strange characters. Okay. So the backslash has to be inside the quotes. Then it's going to be visible. And it's maybe minus E should not have been required. Let's try removing the minus E flag. Yeah, it is still, Vijay, does that answer your question? So you can, there are sometimes you have to play around with your shell and how things work in your shell, but you can have the backspace there. Okay, we are going to carry on now because I need to get somewhere at least with this basic course so that within 2 classes, we are done with the basic command structure. And then I can give you some homework also, hopefully. Yeah, now I wanted to learn about file and directory commands. Up tak we were doing file navigation, basic commands. Now, suppose you want to create a new file. So try this one, the touch command. Try the touch command and then say touch file.txt or touch notes.txt or touch my notes.txt. ऐसा कुछ करो. Okay, let's do this exactly what is written. Touch notes.txt. Let's do this. Can everybody follow these steps and write touch notes.txt? So the touch command basically creates a new empty file for you. You've just touched something and that's a new, and because you've touched it, it's been created. Okay, so let's see what you get with that. Touch notes.txt. What happens when you type touch notes.txt? And I want you to do an ls after that to see whether a new file got created. Let's do it together. Here. So touch. Okay, I want to clear. So the clear command is to clear my screen, right? So I'm clearing my screen and I'm saying touch notes.txt. Nothing happened. Really? Did nothing happen or did something happen? So when I do an ls minus L, I find that in addition to the directory called R, I now have a file. It is a 0-sized file. How do I know it's an empty file? Because its size is 0. So it's been created for all of you. And now what is the next thing I want you to do? So now what you've done is you've created a file, an empty file. So we are going to add some lines to this file. Okay. You all know you just looked at your history. Everybody has some history, right? Now, instead of just printing history like this, you take history and you take history and pipe it into notes.txt. That means the entire history, instead of being printed on the screen, it goes into the file called notes.txt. Is there a
Your
question you
screen,
want to ask?
ma'am, your screen is not appearing, like sharing is stopped.
फिर खो गया. Thank you. ये कुछ drop हो रहा है for some reason. But anyway, I hope you guys know karna kya hai. Kya tha? Terminal. Are you on the terminal? Can you see?
Yes.
All right. So Ashish says that notes.txt बन गया touch करते ही और ls करने पर note.txt आया लेकिन notes.txt आशीष खाली पड़ा है, isn't it? Because उसमें कुछ तुमने सिर्फ touch किया है उसको, तुमने बनाया तो उसमें कुछ डाला नहीं है. So now we are going to put something into notes.txt. You could either type something into like echo something into notes.txt, that will go into notes.txt, but हम पूरा history डाल देते हैं, right? So now history को मैंने डाल दिया note.txt में. Nothing happened. Did anything happen or did nothing happen? Who's going to tell me if something happened and how will I know if something happened? फिर से ls minus l करो और इस ls minus l में और पिछले ls minus l में क्या फर्क था? कुछ फर्क दिख रहा है? Who can tell me? The difference, it is visible on my screen right now. Oh, but is my screen visible? Huh, my screen is visible to you guys now. You see that I did a listing before filling up notes.txt and after filling up notes.txt, and both lines of code basically just tell me, yes, I have two files in my directory. One of them is a file and other is a directory R and notes.txt. तो कोई फर्क नहीं है कि कोई फर्क है, and अगर फर्क है तो क्या फर्क है is the question. All right, Rishabh says all the history commands were texted, but how do you know it? Just by looking at the screen, Rishabh. Subhashish, absolutely right. Radhika, absolutely. Rinshila, you're absolutely right. The file is no longer empty, and I know that because the size is visible in the ls-l command. When you give the minus listing flag to the ls command or list command, minus long flag, it tells you the size, and the size has changed. The size has changed because मेरे पास 10, 11, 12 आशीष की तरह थोड़े सा हिस्ट्री था, लेकिन some of you guys have a big history, तो तुम्हारे फाइल साइजेस बहुत बड़े हो चुके होंगे compared to mine. Is that correct? Anyway, now we have this file called notes.txt and it has some size. Our job is, before we finish today's class, which is till 5 o'clock, I know all of you, we took 2 hours from you. Are some of you willing to join me on Wednesday and let's continue this class on Wednesday? Those of you who feel ki bilkul basic simple hai, you can escape. But others, please join us on Wednesday at 3 to 5 p.m. Just put a yes or no in the command in the chat box to tell me that you can join after we close at 5 o'clock today. In case you can't join, then we won't run this class again. But at least 2 people are willing to join. So, ठीक है. थोड़ा सा ही course रह गया है, but I think it is important that we do it in a DIY. But देखो अभी आपने सिर्फ थोड़ी सी चीजें की है. So let us do one thing. Why don't you all take a little, make this a homework. The homework is what is written on the screen. You have to touch notes.txt. That means create a file called notes.txt जो आप कर चुके हैं. उसके बाद आपने एक directory बनानी है. Directory बनाने का command होता है mkdir, make directory. Make directory, mkdir, और आप वो directory बनाएंगे, उसका नाम होगा my project. Okay? और उसके बाद आप ये जो notes वाली आपने जो फाइल बनाई है, जो अभी खुले में मतलब सड़क पे डाल रखी है, उसको प्लीज एक ढंग की डायरेक्टरी में डालें, my project में मूव करें उसको notes.txt को. Okay? So there is more things. यहाँ पे 5 चीजें हो रखी हैं स्क्रीन पे. सबसे पहले you created a file called notes.txt और उसके बाद आपने एक और काम किया जो इस स्क्रीन पे लिखा नहीं है, जो मैंने आपसे करवाया कि हिस्ट्री को फिल कर दिया आपने notes.txt में. Now I want you to create a new project called new directory called my project, and you will create that directory by using a very specific command which is called mkdir, which is the make directory command. And once you've made that directory, you're going to move your files into that directory. And why this is so important for HPC users, and please note again and again कि ये basic intro course normal Unix का basic intro नहीं है, ये HPC के लिए जो चीजें HPC में आपको use करना ही पड़ेगा. और हम लोग आपका we will be after your life as administrators कि please अपने files को move करो, copy करो, अपनी directory में रखो, common area में नहीं छोड़ने हैं कोई files. For example, my lab home directory is GY lab, and if I have 3 students, you know, Mandeep and Preeti and Renu, and all of them are using, if they create a notes.txt commonly, then I don't know whether it belongs to Mandeep or Preeti or Renu. Even they don't know after 2 days. You guys will forget. So it's very important that you create a directory of your own name inside your lab folder. So within GYLab, there should be a separate folder with the name of each student. Rishabh ka ho, Brishti ka ho, Janvi ka ho, Ritama ka ho, and within that you will move all your files. Either you will do work within that or you will copy and move the files into that, okay? And once you've done this, once you've copied the files, you should remove them from the area where you had created them commonly. So that is why I want you to learn this and get this done. But for now, I want you to just do this one. So we'll close this. We have 2-3 minutes. Why don't we do one thing? 2-3 minutes mein, why don't you ask me something that you have? Or if you think I can add something to the course, then we will add. So basically to the course right now, we finished navigating the file system. Files and directories are going on right now. Iske baad we will do viewing of files. How do you visualize files? And then I will teach you how to start and kill processes. And then we will learn how to change permissions on files. Some files you don't want anybody else to see except yourself. And some files you want everybody else to be able to run and see together. So how do you change permissions? And then I will teach you, well, pipes and redirection I have already taught you with the history, but we will teach you again. Then we will learn some navigation shortcuts, and help also I've already taught you. And then I will give you an end of course challenge. That end of course challenge is something that everybody will have to take, and we can do it on Wednesday. I see quite a few yeses. Please join on Wednesday. Saurav, इसी link को use कर सकते हैं Wednesday को सब students की आप नया link दोगे हमें Wednesday के लिए. I think हमें नया link मिल जाएगा, which Renu I will ask you to share with everybody. And by then, please make sure कि आप लोग अगर NIPGR में हैं और LAN पे हैं, तो आपका DNS sorted होना चाहिए. Okay? So we're going to stop sharing now. And if there are any questions, ask me. There's a little homework. I want you to just practice these commands again and again. And I have also given you a link in your email that this course is built upon so many free courses which are available in the world. But the only difference between this course and what you have done with me is that somebody is holding your hand through it. So it's sometimes useful to have somebody telling you things. That is why we are doing this together. We will do it again on Wednesday and complete this course on Wednesday. But if you have questions, please feel free to ask. Otherwise, Saurav, आप recording बंद कर सकते हैं अभी.